By now, we've learned a lot about inheritance and DNA, but what exactly does DNA do? Well, we'll start this video with a brief overview of how DNA directs the production of proteins and then focus on the first step of that process, transcription. In the early to mid-1900s, researchers were trying to figure out how the hereditary material, whatever it was, could determine the phenotype of an individual. In the 1930s, Beadle and Tatum used a type of mold called Neurospora, and they found that they could induce mutations that changed the way the mold manufactured arginine out of other materials. Now, previous experiments have shown that in order for the cells to manufacture arginine, they needed to have access to citrulline. In order to manufacture citrulline, they needed to have access to ornithine. And in order to manufacture ornithine, they needed access to another precursor. Each step in this pathway requires a particular enzyme. Beadle and Tatum used a variety of growth media. In the first dish, they had standard growth media which contained the precursor molecules. In the second dish, they added ornithine. In the third, citrulline. And in the fourth, arginine. They found that one mutant, here called ARG1, was unable to grow on the standard media, but was able to grow on any of the other dishes. The mutant ARG2 was unable to grow unless citrulline or arginine was supplied to the dish. Lastly, the mutant ARG3 was unable to grow on any media unless arginine itself was added. From this, they were able to map out which mutant strains had which mutations along this biosynthetic pathway. If the mutation was in the last step from citrulline to arginine, then the only way the cells could have access to arginine was if it was supplied directly. If the mutation was between ornithine and citrulline, supplying the precursor or ornithine was not good enough, and either citrulline or arginine was required. Now, each mutant strain, therefore, had a mutation in a different gene, and when a gene was mutated, it disabled the biosynthetic pathway at that step. This finding gave support to an important theory that each gene was responsible for the manufacture of a particular enzyme, and this was known as the one gene, one enzyme hypothesis. Even though this was a very useful hypothesis at the time, we now know that genes make things other than enzymes. For example, all proteins are made from genes, and RNA molecules that may never be translated into protein are also made from genes. We also know that more than a single gene may be required to make a functional enzyme or protein. This is the case particularly when the quaternary structure of the protein involves two separate polypeptides. Finally, we also know that a single gene may make more than one type of protein through processes such as alternative splicing, which we'll learn about in a separate lesson. As we've learned before, DNA can be seen as a repository of information. This information is not very useful by itself, and must be used in order to make other things for the cell. In the manufacturing of proteins, or more generally polypeptides, the DNA is transcribed into messenger RNA or mRNA. The mRNA is then translated into a peptide. One way to help you remember these words is that DNA and RNA are both nucleic acids. In a sense, they're both different forms of the same language. For example, if I say something and you write it down in the same language, then you're transcribing my words. Similarly, mRNA is a transcription of the same code in DNA form. Polypeptides, however, are like a totally different language, and so to get from mRNA to polypeptides, you need to translate. There's an important difference between bacteria and eukaryotes. Bacteria don't have nuclei, and so transcription and translation all happen in the same space. In eukaryotes, however, the DNA is stored safely in the nucleus, but polypeptides are formed in the cytoplasm. Transcription happens in the nucleus, and the mRNA transcript is then modified in a series of processing steps before it's exported from the nucleus into the cytoplasm, where translation occurs. Here's another way of looking at that. Again, a particular gene on DNA is transcribed to form a transcript of messenger RNA that's essentially the same type of language. The code on that molecule of mRNA is then read in order to create a polypeptide where the linear code on the mRNA is directly translated into a linear string of amino acids. Translation is a pretty complex process. However, researchers have worked out the exact code on the mRNA in order to join together particular amino acids into a polypeptide. We'll look at the process of translation in another lesson. For now, however, let's look at how mutations at the DNA level affect an individual's phenotype. At the top here, we have a DNA sequence that's part of a gene that determines the coat color in mice. That code gets written into the form of mRNA through the process of transcription. Note here that, and we'll come back to this, that the code written onto the mRNA is exactly complementary to the code in the DNA. The only difference here is that RNA does not use the base thymine. Instead, a related base called uracil is used, and uracil is complementary to adenine in the same way thymine is with DNA. This mRNA is then translated into protein, which may be an enzyme that gives the mouse a dark coat color. In a different mouse, the DNA sequence is slightly different, having an adenine at this first base instead of a guanine. As a result, the mRNA is also different, and that code is then used to create the polypeptide, which is also different. This particular polypeptide, in turn, causes this mouse to have a light coat. As was the case with replicating DNA, transcription also requires a polymerase. However, in transcription, that polymerase is an RNA polymerase, named because it's polymerizing RNA. Also, like DNA replication, the RNA polymerase also moves in a 5' to 3' direction, adding bases to the 3' end of the RNA transcript. Specific sequences along the DNA strand are identified by the RNA polymerase. In particular, these sequences are generally known as promoters. In bacteria, there's an additional protein called sigma that binds to the RNA polymerase. Sigma is responsible for identifying the promoter. There are particular sequences of DNA that sigma identifies, and these are known as the -35 and -10 boxes. The numbers come from the numbering scheme of the RNA itself, with the first base of RNA being base number 1. Therefore, bases that are upstream, or before the bases that are transcribed, are numbered with negative numbers, and numbers that are downstream of those that actually get transcribed are positive. In any case, sigma identifies the promoter sequence and determines the correct orientation of the enzyme in relation to the DNA strand. Binding to the promoter also determines which strand of DNA gets transcribed. Remember that the other strand of DNA is not the same, but complementary, so that particular sequence only occurs on one strand. As initiation completes, the RNA polymerase unwinds the DNA, separating the two strands and allowing elongation to start. In eukaryotes, the process is broadly similar. However, eukaryotes have several RNA polymerases, each responsible for transcribing certain types of RNA. RNA polymerase II is responsible for making mRNA. Eukaryotic promoters are also more variable than bacterial promoters. Many of these, however, include a sequence of TATA, known as a TADA box, and this is also upstream of the transcription start site. Lastly, the binding of the RNA polymerase is directed by the presence of transcription factors, molecules that bind to the DNA and identify the sequence as the promoter. Once the DNA has been opened up, elongation can start. The RNA polymerase moves downstream along the DNA strand, and RNA nucleotides are added to the growing RNA strand. As with synthesizing DNA, the RNA nucleotides are triphosphates, allowing the addition of new nucleotides onto the 3' end to be exergonic and spontaneous. The extra phosphate groups are cleaved from the new nucleotides to form the sugar phosphate backbone of the RNA once the new nucleotide has been determined to be complementary to the template DNA. As the polymerase moves downstream on the DNA, the RNA molecule peels away from that DNA template, and the two strands of DNA then reform the double helix. Let's take a look at the names of these two DNA strands that these two DNA strands are given, since this causes a lot of confusion for students. The most important strand here is the template strand, and that's easy because it simply serves as a template for creating the RNA. The other strand is called the non-template strand, which is also easy. However, the non-template strand is also often referred to as the coding strand, even though it has nothing to do with coding for the RNA. The reason it's called the coding strand is that the code on this strand is exactly the same as the code on the RNA. This is because the coding strand and the RNA are both complementary to the same template strand. When the RNA polymerase has completed transcribing the gene, transcription terminates. In bacteria, the signal to terminate transcription is intrinsic to the mRNA that has been produced. The mRNA forms a hairpin loop, and the loop, in coordination with particular sequences that is in the particular sequence that's currently in the polymerase, causes the polymerase to pause and may terminate transcription. Another mechanism, called Rho-dependent termination, involves a protein called Rho identifying the termination signal and causing the polymerase to stop transcribing. In eukaryotes, a polymerase continues transcribing past a polyadenylation or poly-A signal. This poly-A signal becomes part of the mRNA transcript, and at some variable point downstream of the poly-A signal, the RNA polymerase falls off the template strand, terminating transcription. In prokaryotes, once transcription is complete, the mRNA transcript is available to be translated into protein. This is because prokaryotes don't have a nucleus that separates transcription from translation. In eukaryotes, however, the mRNA needs to get from the nucleus into the cytoplasm for translation. First, however, the mRNA needs to be processed in a number of ways. The first part of RNA processing involves RNA splicing, and this starts happening while transcription is still going on. Eukaryotic genes have sections which will be removed from the final mature mRNA transcript. These portions that are removed are called introns, and the portions that remain and are exported to the cytoplasm for translation are called exons. In most genes, the bulk of the gene is actually intron sequence. For example, the gene SHANK3, which is involved in neurotransmitter function and has been implicated in an autism spectrum disorder, is a gene of about 60,000 base pairs of DNA. However, most of this is intron sequence and is cut out, leaving only about 5,400 base pairs of the mRNA. And this is actually spread out over 23 different separate exons. The work of splicing out the introns is done by a complex called the spliceosome, which is a complex consisting of over 300 proteins and several different RNA molecules. The RNA molecules are used for identifying the splice sites by their sequence. Once located, the proteins bring the ends of the exons together, cut the DNA, and splice together the exons so that the resulting mRNA is a continuous transcript sequence that will be translated once it arrives in the cytoplasm of the cell. After the introns are removed, they quickly degrade in the nucleus, and the nucleotides will eventually be reused. There are a number of really interesting evolutionary implications of this type of genetic architecture. In some cases, genes are constructed in a sort of modular way, with introns corresponding to functional or structural domains of the resulting polypeptide. This allows for new genes to be created by a sort of mix and match method called exon shuffling. Additionally, exons can be joined together in different combinations to produce varied polypeptides. In some cases, the exons can be cut out along with the introns. Introns can be retained, or there might be certain combinations of exons that are mutually exclusive of one another. All these mechanisms combine to create the possibility of forming many different polypeptides, all with a single gene, an obvious departure from the one gene, one enzyme hypothesis. The last two things that need to be done are the addition of a poly A tail and a 5' cap. Both of these modifications are necessary in order for the ribosome to bind to the mRNA to initiate translation. And they're also needed to protect the mRNA from nucleases, enzymes that can break down nucleic acids that are present in the cytoplasm of the cell. Once the introns have been removed, the poly A tail and the 5' cap have been added. The mRNA is then ready for export into the cytoplasm and to be used as a code to create polypeptides.